Revision, steps in tissue culture, practical applications, and the nutrient medium. Plant tissue culture is a technique of growing plant cells, tissue, organs in an artificially prepared nutrient medium under aseptic condition. Tissue culture techniques involves the following steps: selection of a suitable plant, selection of suitable part of the plant, that is explant, culture of the tissue, propagation of the plant, and then transportation. First, coming to selection of suitable plant. This is the first step. A plant which is economically important, that is, it may be with regarding to medicinal value, food value, etc., but fail to produce seeds, or seed setting is low, or the life cycle is very long, as in case of bamboos, can be selected for tissue culture micropropagation. Even tree species like teak, sandalwood may also be selected. Second one, selection of suitable part of the plant. Selection of suitable part for the purpose of culture. A suitable part of the plant to be propagated may be selected. This may be a piece of tissue, stem, axillary bud, embryo, seed, or even pollen grain. Culture of the tissue. The tissue selected for the purpose must be separated from the plant. This is now known as explant. This explant must be washed thoroughly to remove the contaminants and placed on a nutrient medium. A suitable culture media is prepared with special attention towards the objectives of culture and type of explant to be cultured. Prepared culture medium is transferred into sterilized vessels and then sterilized in autoclave. The sterilized explant is inoculated on the culture media under aseptic conditions. The cultures are then incubated in the culture room where appropriate conditions of light, temperature, and humidity are provided for successful culturing. Cultured cells are transferred to a fresh nutrient medium to obtain plantlets. Then propagation of the plants and transplantation. Plantlets growing in tissue culture tubes or flask are very delicate and can be transferred to tiny pots and kept in protected greenhouses. This process is called hardening. After the plantlets are several weeks old in the greenhouse, they are transplanted to the normal pots or to the field for further growth. Coming to the practical applications of tissue culture. First one is the germplasm conservation, mainly in the form of cryopreservation of somatic embryos or shoot. Second, the technique of micropropagation for enhancing the rate of multiplication of economically important plant species. And third is eradication of systemic diseases in plants and raising disease-free plants. The next one is soma-clonal variations or useful source of introduction of valuable genetic variation in plants. plants. And it also helps plants in imparting resistance to antibiotics, drought, salinity, diseases, etc. Somatic hybrids and hybrids overcome species barriers and the sexual incompatibility and produce hybrid plants with desired combination of traits. Embryo culture helps in overcoming seed sterility and dormancy. Haploid production in culture helps to solve various problems of genetic studies and therefore help the plant breeders for producing new varieties. Coming to the nutrient media. The basic components are some of the important nutrient media are MS medium, that is Morashiki Shug medium, LS medium, B5 medium, White's medium, etc. Important constituents of a culture medium are organic supplements, that is vitamins like thiamine, pyridoxine, nicotinic acid, etc. Then antibiotics like streptomycin, kanamycin. Amino acids like arginine, asparagine. Inorganic nutrients. Micronutrients such as iron, manganese, zinc, molybdenum, copper, boron. Macronutrients include 6 major elements such as the nitrogen, sulfur, phosphorus, potassium, calcium, magnesium, carbon, and energy source. Most preferred carbon source is sucrose. Others include lactose, maltose, galactose, raffinose, cellulose, etc. Then growth hormones like auxins, mainly for inducing cell division, cytokinins, mainly for modifying apical dominance and shoot differentiation, then abscisic acid and gibberellins are used occasionally. Gelling agents. These are added to the media to make them semi-solid or solid. Agar, gelatin, alginates are the common solidifying or gelling agents. Other organic extracts. Sometimes culture media are supplemented with some organic extracts like coconut milk, orange juice, tomato juice, potato extract, etc. Types of tissue culture. One is callus culture. It can be defined as production and maintenance of an unorganized mass of proliferative cells from isolated plant cell, tissue or organ by growing them on artificial nutrient media in glass vials under controlled aseptic conditions. Anther culture. Androgenesis is the in vitro development of haploid plants originating from potent pollen grains through a series of cell differentiation and cell division and differentiation. Pollen culture. It is the in vitro technique by which pollen grains, preferably at the microspore stage, are squeezed from the intact anther and then cultured on nutrient media where the microspores without producing male gametes. Embryo culture. Embryo culture may be defined as Aseptic isolation of embryo from the bulk of maternal tissue of mature seed or capsule, and in vitro culture under aseptic and controlled physical condition in glass vials containing nutrient semi-solid or liquid medium to grow directly into plantlets. Thank you.